Hello, my name is Sofia Saavedra. I, along with Eduardo Santiago, am a computer engineering student from the University of Puerto Rico at Mayagüez working on this project. I will be presenting our project on the effectiveness of in-situ microbiotic observations via an embedded marine operating, sensing, and imaging stereoscope. Dr. Manuel Jimenez is the advisor for this project, along with co-advisor David Repollet. Some of the most important activities that impact our oceans and coastal ecosystems occur in a microscopic scale. Some examples being coral bleaching, competition between organisms, and fluxes of particles to and from the seafloor. However, observing these important events in their natural environments is a challenging task. In this project, we propose observing the attachment of local wild cultivars of benthic algae onto a 3D printed varying topographical growth plates in two ways, in situ or in place and in a lab environment. The goal being to contrast the effectiveness of conducting this algal recruitment experiment with an underwater stereoscope prototype, which will provide in situ monitoring of the surrounding water quality as the establishing algae begins to develop. Data from both methods will be collected and studied at a future occasion in the laboratory while retaining their raw in situ characteristics that were present at the moment of conducting the sampling process. This data can in turn be used to correlate the visual behavioral component of the organisms being observed while taking into consideration how key in situ ambient variables are affecting them. There are two main problems presented in this project. As mentioned previously, some of the most important activities that impact our oceans and coastal ecosystems occur in a microscopic scale. As such, these events are important and of great interest for research. However, one cannot easily replicate the complex and ever-changing conditions of these natural systems in a lab, as organisms lose valuable characteristics once they are removed from their environments. Thus, the only option to fully capture and study these environments reside in situ research. Unfortunately, even though many existing laboratory instruments benefit the researcher in situ or in vitro, few work in the marine environment. Even if the instruments are waterproofed, they represent a cumbersome load on the marine research diver. To solve this, we propose MOSES. The Marine-Operated Sensing Imaging Stereoscope, MOSES, will be a device capable of capturing stereoscopic images with a log of key sensor readings from the surrounding ambient water. The device itself shall be easy to manipulate via scuba diving, in which a researcher places the device over or looking towards the selected sample area. This device will be separated in two units, an optical unit and a sensory unit. The optical unit will be comprised of two microscope cameras to create stereoscopic images, a 3 lightning control mode to capture the image in near-infrared, white, and near-ultraviolet light, and a user interface to control camera and system settings. The sensor unit will include a thermometer, barometer, pH, and dissolved oxygen sensors. The integration of sensors into the microscope is aimed at reducing the % of human error in experiments conducted in the marine environment, where portable means of imaging and frequent water sampling are needed. By automating part of the sampling procedure and sampling sensory data along with image data, the imaging device can provide a stable means of observing and correlating small sessile benthic organisms in their natural environment without removing them and altering experimental results. The main project objective is to assess MOSES suitability for in-situ underwater studies. Reaching such an objective requires two sequential sub-objectives, those being delivering a functional MOSES prototype and performing assessment tests in an underwater environment where benthic algae attachment can be observed. To complete the first sub-objective, our team will need to complete a MOSES prototype that includes a submersible stereoscope that can retrieve microscopic underwater images and environmental data that complements the microscopic pictures. The second sub-objective is performing actual in-situ tests with the instrument prototype to assess its fitness for underwater studies. The first phase of this project is aimed at completing the first sub-objective, the delivery of a functional MOSES prototype. First, we will integrate a camera module system consisting of 2 microscopic cameras and a LED light ring. The module will be developed around a Raspberry Pi 4 microcontroller. Second, we will implement a sensory module that incorporates a temperature, pressure, dissolved oxygen, and a pH sensor. This module will be developed around a Texas Instrument TIVA microcontroller. Third, we will implement a user interface to control the camera and sensor modules and offer some camera features such as image capture modes and sensory data retrieval modes. Following, we will implement a communication system between the sensor and camera modules. And lastly, we will integrate the whole system together. Here we can see the global design of the system, separated in two modules. As mentioned, the microcontrollers used for this project will be a Raspberry Pi 4 working as the primary controller and a Texas Instruments TIVA Series C microcontroller as the secondary microcontroller. The second phase of this project will be aimed at completing the second objective: perform assessment tests in an underwater environment. We will first compare the data recovered by leaving two sample plates exposed to an open water column for a week's time. Plate 1 will be monitored via traditionally available lab equipment, while Plate 2 is monitored with the MOSES prototype. In both cases, the same type of sample plate will be used for both tests and observed for the same amount of time. After, the sample plates shall be recovered from the water and observed to determine the percent of colonization and possible algae types present. Final results will then be extracted based on observations made with both methods. So far, we have succeeded in completing great part of the first objective to create a working prototype. For future work, we will need to encase the final MOSES prototype into its submergible vessel to test underwater. This includes thorough testing before deploying it in an ocean environment. Also, as phase 1 of the project is close to finishing, we must then start phase 2 of the project, the underwater assessment.